So let's discuss comparative embryology. Now, we've discussed looking at a lot of adult characteristics to try to compare them, to try to see if we can discover these vestigial traits, homologies, analogies. We can analyze the fossils to try to do this for past life. But one of the other cool avenues to try to notice these homologies is to look at the embryos. Because when you're developing as an embryo, you go through many phases that disappear before you're born. They don't show up in the adult organism. But we can compare these phases that you go through to embryos of other organisms to try to see if you're related, to see these, embryonic homologies. And we call that field embryology or comparative embryology. So if you look at humans, you'll see that we have, when we're developing, these pharyngeal slits. And so these in us develop into parts of like our jaw. You can have where some become parts of the inner ear. Whereas in fish, these become gills primarily. In reptiles, they become part of the jaw as well. So these can become different things later on. But we still see that we all have the same structures early on in embryological development, when it's not like we have to have pharyngeal slits to ultimately get a jaw. We can see that humans, just like pretty much all other vertebrates, have a tail when they're an embryo. Now, they can lose this tail as you move on, like we do, as other apes do, but ultimately we still see the evidence that it's in our genome. We can still see embryologically that we share this characteristic with the other vertebrates, so it helps link all those vertebrates together. There's additional things, how the kidneys develop, how the circulatory system itself develops. So there's lots of different traits that we have as an embryo that we lose as an adult that can still show us this homology, this shared ancestry. Comparative biochemistry is where we can now analyze the proteins, the DNA, the RNA that we have in our cells, and we can compare that with other species. And the basic idea here is if we were one species at one time with another organism, so this is a common ancestor, so we're looking at, let's say, this guy right here. So when this particular organism split into two populations that eventually became two separate species, it should have had most of the same proteins and most of the same DNA and RNA, because they recently evolved from one species that had the same DNA, RNA, and proteins. But as time passes, we'll get more and more mutations, which means we can get more and more new genes. So the DNA modifies, new variations of the RNA, new types of proteins. And so we see as time passes this exact same characteristics. So if we're managing to track these kinases, which is protein kinases are a type of enzyme, as we track them, we can see as they start to become more and more diverse, and we can see as they become more and more different from each other. And so this allows us to then figure out how closely related two organisms are by tracking the similarities between their genes, between their proteins, between their RNA. So if you were going to compare our protein kinases to those of other vertebrates, you'd see many similarities. But if you tried to compare ours to early organisms, like the opisthecons, are basically the guys that really first started out animals in general, you'd see there's some similarities, but not near as many as there'd be with, modern vertebrates. So if you compared us to like a dog, we'd have a lot more similarities than if you compare us to a protozoan that's about to become, a sponge or something like that. So this fits the mold of what we expect with evolution, and we use this to track who's more related to who. This is kind of cool too, because we didn't have this evidence back when we were first setting up a lot of these relationships. When Darwin was doing his work, we didn't know about genes, we didn't know about, DNA. And so now that we have it, this has been a great way to check our work, because we can make predictions without having been able to know this, and then we can test it and verify that it's occurring exactly as we expect it to. And so this strongly supports the idea of evolution, because otherwise there's no reason to see this pattern where things that appear to be more closely related to us based upon fossils, etc., would also have to have much higher degrees of relatedness in their DNA, RNA, and proteins. Another cool side effect of biochemistry is we can look at certain genes and we see that they have about a constant-ish rate of mutation. It's not perfect, but we have a general sense of about how often mutations occur over the course of time. And so if we know about the mutation rate, how often these mutations occur, and if we know the number of mutations that have occurred between two organisms that we think were related in the past, we can then effectively calculate when that divergence occurred, when they split from one type of organism, one common organism, into two separate populations that evolved their own separate ways into their own separate species. And so this is kind of cool for us to track back either within a species, like humans, about how long ago blue eyes occurred as a mutation, because we can do that even within a species, just by calculating mutation rate and ultimately what's going on with the number of differences that had to occur for us to get this trait. And we can also do this to try to figure out, like us and chimps, about how long ago this occurs. So this is a great way to help verify a lot of our other dating mechanisms, such as relative dating, absolute dating, tidal dating. There's lots of them. So it's just yet another way for us to feel comfortable with the chronology as we're doing stuff. Now, many genes, and the way this works best, is to look at ones that change rapidly. So these are called poorly conserved. But you can also use ones that are highly conserved, which means they change very slowly, like our RNA genes, the ones that make up your ribosomes. They change very slowly. And so these can be used even if you want to check things that go further back. But because this is an approximate thing, the further back you go, the more of a margin of error you get. So we prefer to use this for things that are more recent. It just tends to work out better, and it tends to be where we get a better ballpark. We feel more comfortable with it. Now, our last type of evidence for evolution that we're going to discuss is biogeography. Now, biogeography is the study of where organisms live and essentially trying to understand why it is that they live there. Now, when we look at evolution, we can see several interesting bits with biogeography. The first is that we tend to see species that are very similar on the mainland, and we tend to see very similar species on islands that are nearby. So it gives us this idea that the species on the islands appear to have originated from the mainland. You'll also see that species on islands that are nearby will oftentimes be similar to other islands. So if you have a chain of islands, you might see 15 different species of the same original mainland species. So like with Darwin, he might see lots of species of different finches, because it appears as though that some finches from the mainland managed to migrate and get their way out there, at which point, as they move to different islands, because those are pretty nearby, they would be in slightly different environments, they'd be reproducing only with the other ones that are on the island, and they'd have slightly different mutations that occur. And so as a result of this, we'd get a different species that develops over time on each of the islands. So we get lots of species of finches. Now, this connectedness between mainland species, between island species, and the difference that we see between species on different continents is kind of critical. Because, for instance, if we have something like a rabbit, which is what we call a placental mammal, keeps its young all the way inside until birth when they're pretty much good to go. And so these guys live on most continents, and they're very successful in grassland areas that are open. But when we look at Australia, we see somebody that looks kind of like a rabbit, but it's analogous. These are marsupials. They have many of the adaptations that are similar to rabbits because they live in a similar environment, but they're not actually rabbits. Why would this be? If you've already got rabbits that are well suited to this grassland environment, why wouldn't they be everywhere where it fits? It doesn't make sense that we'd go through and make something else exist that's very similar to a rabbit, but it got there completely separately. We've essentially designed a semi-rabbit multiple times. Why do that when we already had a rabbit? So this doesn't really make sense if you're placing things places, but it makes perfect sense if you just say, rabbits appear to have been able to reach certain environments by migrating, by moving, but they couldn't get to Australia. And so in Australia, something else kind of gradually was selected for and evolved to fill that niche. But it couldn't be a regular rabbit because they weren't there. You had to use something else originally, in this case, marsupials. Now, we also see some interesting things when we look at coastlines, where on different continents, at certain points in history, we'll see similarities in species on these coastlines. Now, this is best explained by plate tectonics, because the particular continents that we have not always been where they are now. We've got these large floating crust chunks that are floating on top of this churning mantle that because of heat gets these currents, and so they gradually move along at probably half an inch to maybe a couple inches per year. But over large periods of time, this can be significant movement. So you might remember back that a long time ago we had Pangea, so around 250 million years ago, all the major continents were essentially stuck together. Now this is interesting because that means species that were living in many of these areas where the continents separated, currently it was together. So those species were just living there and they had no sense of are they in South America or Africa or North America. They were just living on contiguous chunks of land. So those species we see at that time interval are the same species. We don't see a difference between the essentially eastern coast of South America and the western coast of Africa species. But then as time passed, we saw these continents shift away from each other. So these populations got separated, and then they underwent their own separate mutations and natural selection, and they eventually became different species. And we can see over time that this is what occurs. We start to see that there's these differences, this divergence that occurs on these separate continents now that they've split up. So this perfectly fits our idea that most of the stuff going on is not due to placement, it's due to migration. And this migration has fluctuated over time because we have changing climates that occur in large part due to plate tectonics, this shifting of the Earth itself. A great example of that is we talked about those Australian marsupials. If you go back in time, you'll see that marsupials evolved in South America, it appears, but then they migrated through Antarctica, which at the time was much warmer, was much more lush, and eventually into Australia, which was on this eastern side, and broke off later. You can't kind of see it swinging out in this picture, but it would be there. And so these marsupials gradually migrated through these three attached chunks. But then eventually South America split away and hooked up with North America via Central America here, and placental mammals were able to come down and outcompete most marsupials. Not all, but most of them. You'll see Antarctica got to be wicked cold, and so that basically killed off everything. But Australia, that was kind of hanging out over here, that one separated, still kind of full of marsupials. And because placental mammals have never been able to get there until modern times with humans, they were able to evolve to fill all these niches that were available. That's why we see, kangaroos there. That's why we see sugar gliders that look like little flying squirrels, but they're marsupials. They're not actually flying squirrels. So everything was more reptile and marsupial based there because of this isolation that occurred. But it originally got marsupials due to this migration ability. Doesn't appear they're placed there. Instead, in the fossil record, we see this gradual shift as they kind of migrate, and we see that they've managed to hang out in Australia now until today.